thanks to the organizers for this great opportunity. Can everyone hear me? So today I'd like to talk about a recent project, unpublished work on mapping the evolution of T cell states during response or resistance to a form of immunotherapy called DLI. And this work is in collaboration with Pavan Bhattacharjee, a clinician scientist in Kathy Wu's lab at Dana-Farber, of course, my mentor, Donna Peer, and the computational modeling that I'll talk about is joint work with Cassandra Burziak and Zining Chu, two grad students in the Peer lab. So to give you some background, in myeloid malignancies, patients receive, a standard of care is to receive a hematopoietic stem cell transplant. And in 25 to 40% of the cases, the patients relapse after transplant, and at that point, the median survival is around 6 months, and the treatment options include additional chemo or immune suppression taper, a second transplant, or receiving an infusion of lymphocytes from the original donor called donor lymphocyte infusion, or DLI. And this is an established type of immunotherapy that has been given to leukemia patients for decades. And because of that, we have the great advantage of access to well-annotated clinical cohorts with paired bone marrow biopsies before and after therapy. So we decided to look into DLI as a clinical model to dissect anti-tumor immunity. So what is known about DLI? Our collaborators at Dana-Farber have previously shown that the patients that respond to DLI have a more enriched T cell exhaustion before receiving the treatment, and they also see a reversal of T cell exhaustion after DLI. And this has also been seen in multiple types of leukemia, CML, AML, CLL. Although we know about this reversal of T cell exhaustion, what is unknown is what exactly are the T cell subsets that mediate this reversal of T cell exhaustion. And in general, what are the T cell mechanisms that mediate response or resistance to this type of immunotherapy? So the questions in our study were, can we have a systems approach to find where the underlying factors that explain the global phenotypic diversity of T cells in the context of DLI, and also how do these T cell states evolve, or how are they impacted by this treatment? And then what are the unique features of T cells that potentially contribute to a response or resistance? So the experimental design involves a cohort of chronic myelogenous leukemia patients, CML, and these patients have previously received a transplant and then relapsed. And so a subset of our patients after receiving DLI show a response, meaning decrease in tumor burden, and a subset don't show a decrease, meaning don't respond to the treatment. So for each patient shown with lines in this figure, we have previously collected bone marrow biopsies at multiple time points before and after the treatment. And we also additionally, as control, had 2 patients that did not relapse after transplant. From each patient time point, we collected both single-cell RNA-seq, which has been mentioned in a few talks today already, and attack-seq data, bulk attack-seq data on sorted subsets of CD8 positive or CD8 or 4 positive effector memory or central memory T cells. So using our previous probabilistic modeling approaches, in particular cell type specific normalization method called BISCUIT that was published last year, we identified 65 unique T cell states shown here in a T-SNE map where cells are colored by clusters from collecting 97,000 T cells that passed our quality control metrics. And you can see that these T cell states are shaped by the timing of the therapy shown in pre versus post therapy and normal, but are also showing associations with the outcome in different proportions of T cells seen in responders versus non-responders in blue versus orange in this map. So our first question was, can we find, using a systems approach, what are the underlying factors that explain the global phenotypic diversity? To answer this question, we used a technique called common factor analysis. And to illustrate why we decided to use this method rather than other types of factor analysis methods, let's assume we have two sets of cells corresponding to two trajectories, let's say activation and exhaustion of T cells, that could correspond to the same response mechanism in this case. And these mechanisms or trajectories could be correlated but not collinear. Now, if you try to do PCA or diffusion component analysis and generally factor analysis methods that try to explain maximize the explained variance, you will get different components for these two trajectories. But if you do common factor analysis, this method assumes that there are underlying latent factors that explain shared variance, shared components of variance, and therefore covariance of cells along certain dimensions. And therefore, it ignores the proportion of variance that could be unique to the cells therefore, the factors themselves, unlike PCA, are not necessarily a linear sum of the common factors. So applying this method on our data, we found three major factors, common factors, that showed a separation of T cells enriched in non-responders from T cells enriched in responders. And when we annotated the factors, we found factor 1 that was associated with responders, in addition to exhaustion, which was known before, was also enriched for T cell activation, interferon, glycogen metabolism. And factors 2 and 3, which were associated with non-responders, showed enrichment in different forms of T cell dysfunction, such as peripheral tolerance, hypoxia, T cell anergy, deletional tolerance, etc. Now, what was interesting was that the response mechanism, the enrichments is shown in factor 1, was mostly seen in all of the responders. However, the different dysfunction mechanisms that we see in non-responders showed differences, significant differences across non-responders, as shown in the distribution plots in this map. And this suggests that there could be multiple forms of resistance mechanisms rather than compared to a shared response mechanism in responders. And these different forms of dysfunction mechanisms in non-responders also reminds us of the Anna Karenina principle that all happy families are alike, each unhappy family is unhappy in its own way. So now our main question was how are the T cell states impacted by therapy? To answer this, first we did a quantification of the global diversity of T cells using a metric we devised called phenotypic volume. This metric measures the pseudo-determinant of the gene-gene covariance matrix, and therefore it increases with a number and degree of independence of components that explain the modules of co-expressed genes. When we applied this, surprisingly, we found that the T cell phenotypic diversity at the baseline level, meaning before receiving the treatment, in the responders was much higher than that seen in the non-responders, suggesting that there could be specific T cell states, diverse states that are lacking in non-responders and are important to the response mechanism. Surprisingly, though, both groups of responders and non-responders showed a significant increase in the diversity after treatment. So the non-responders don't show a clinical response, but still undergo a significant remodeling in terms of the transcriptional diversity. So we've looked at this globally, but what are the unique features of T cell states that contribute to the phenotypic expansion that we see being induced by therapy? So to answer that, we first looked at a group of samples that had more consistent timing. So a first group right before receiving the DLI, right at day 0 or around 10 days before, and a group of time points around 300 days post-therapy. And then we looked at the distribution of the T cell clusters shown in columns in this dot plot across these clinical groups, responders pre, post, and non-responders pre and post. And the radius of the circles is proportional to the distribution. So first we found multiple T cell states that are that are predictive of outcome at the baseline level, but surprisingly, none of the non-responder specific states were consistently seen across all patients. Again, kind of supporting the Anna-Karenina principle, but 4 of these T cell states enriched in the responders were consistently more abundant in responders compared to non-responders, and these were all CD4 positive clusters. And in identifying these clusters, we actually used an unequal variance t-test because the abundance of each cluster in each patient has a different sensitivity because we collect different numbers of cells in each sample, and therefore the probability of detecting a particular abundance is different across samples. Now, what happens after treatment? So we first see a diverging pattern of T cell states between responders and non-responders, meaning the distances between the clusters and the diverse phenotypes increases between the 2 clinical groups. And this increase and expansion in the responders is associated with pro-inflammation, interferon activation, and in non-responders, of course, is associated with resistance. We identified 2 main sets of clusters, T cell states that expanded in almost all of the responding patients, but did not show any significant change in non-responders. And again, this consistency shows that the responder patients should have common shared pathways of response. And another interesting observation was that these sets of clusters actually are mixed lineage. They're a combination of CD4 and CD8 T cells, unlike the baseline predictive T cell clusters, which were only CD8 positive. Now, in both the expanding T cells and the baseline enriched T cells, we saw a lot of hits for enriched T cell exhaustion. So this motivated us to dig deeper into doing a better job at annotating them and finding out what's the difference. We actually found different subsets of exhausted T cells, especially in recent studies, showing that there are these progenitor exhausted T cells that are polyfunctional, and over time they have the ability to differentiate into terminally exhausted T cells. However, studies in mice have shown that only the progenitor exhausted subsets respond to PD-1 blockade treatments. And when we use these studies to curate our own signatures and apply this to our clusters, we found, interestingly, that all of the clusters that were enriched at baseline in the responders were all terminally exhausted, whereas the ones that are expanding over after treatment in the responders are all progenitor exhausted cells. So we also confirmed some known markers such as TCF7 in the progenitor exhausted, and also found novel markers for these clusters. So now this kind of motivated us to look more into the association relationship between these 2 types of exhausted T cells. And one question we had was, are there shared underlying mechanisms and shared regulators between these subsets as well? So for this, as I mentioned, we collected ATAC-seq data as well. When we started this project, there was no single cell ATAC-seq, so we did a bulk ATAC-seq on sorted subsets of T cells. So each subset of, let's say, CD4 positive effector memory is associated with multiple clusters of T cells that we see in our single cell RNA-seq data. So we had to develop a method that not only deconvolves the bulk ATAC-seq profile, the accessibility profiles to cell type specific profiles, but we also wanted to combine these with the single cell expression data, specifically the co-expression patterns between genes to infer gene regulatory networks that are specific to cell types. And the way we did this was developing a method with Cassandra Borziak called Symphony, which is a Bayesian multi-view mixture model. I'll briefly go over the generative process. Let's say we have 2 different cell types in our system. Each cell type has a latent accessibility profile that we want to infer. From these, we observe our bulk ATAC-seq profiles. Our main assumption is that the bulk ATAC-seq profiles are a weighted sum of the cell type specific profiles, with the weights being the proportion of the cell type in the sample. And we assume that we also have paired single cell RNA-seq data from the same sample. Combining these accessibility profiles with motif information in the accessible peaks, we can have ideas of predicting what transcription factors could potentially be regulating target genes. And the function, meaning activation or repression, and the strength of the regulation could be informed through the co-expression patterns between the transcription factor and the target in different cell types. Which is seen in the single cell RNA-seq data. So we have these 2 observations of ATAC-seq and single cell RNA-seq, and these 3 parameters are the main latent variables that we're trying to infer. I won't go through the details of the scary looking model. You can find that in the archive, but we provided a variational EM inference and scalable implementation with Edward for this model. Applying it to the data, what was really surprising was we found that, first of all, the architecture of the regulatory networks is very different even across the terminally exhausted clusters. But we also found, for example, TOX regulators or TCF7 acting as hub regulators in driving genes, especially genes markers of terminally exhausted subsets. So this kind of shows that the underlying mechanisms of the progenitor exhausted subsets and the terminal exhausted subsets have some commonalities in terms of the master regulators. So this, again, motivated us to now think of, okay, how do these subsets look relative to each other in terms of the full dynamics and the evolution of the T cells over time. So for that, we wanted to pull in the other samples that we had excluded in the previous study. Now, the problem with these samples, so these are all the pre and post samples. When we want to model the dynamics of how the proportion of T cells in a particular cluster changes over time, we encountered two challenges. The first was the timing of the samples. As you can see there, this is just a cartoon showing the relative times, but they're not consistent across all patients. But also, as I briefly mentioned before, the sensitivity of detecting abundance is very different across samples. So our uncertainty in measurements is different due to different sample sizes as well as cluster sizes. So to tackle the first challenge of the non-uniform sampling, we used the Gaussian process regression model that models the dependency between all pairs of time points. And then to tackle the second challenge of variable cluster and sample size effects, we adapted the Gaussian process to weigh the observations based on the uncertainty through a prior that is driven by the number of cells detected. And we have this also a variational inference scalable implementation in Pyro for this model. Now, applying this model to the data, we saw that the terminally exhausted subsets, the proportion of these T cell clusters in responders shown in blue here, nicely follows the tumor burden dynamics that we see in the responders, which go down after receiving the treatment, whereas they do not follow the tumor burden in the non-responders. And the interesting dynamic in the progenitor exhausted subset was that we see this expansion much later after the significant abundance that we see in the terminally exhausted subsets. So the summary so far, what we've understood from this data is that in the responders, we saw an enrichment in the terminally exhausted subsets at baseline. And we see the expansion of the progenitor exhausted subsets induced by the therapy. And in the non-responders, we see different forms of the T cell dysfunction, which are also heterogeneous across patients. Now, one question we had was, where are the progenitor exhausted T cells coming from? Are they coming from the donor lymphocyte infusion, the immunotherapy that is given to the patient, or are they really just expansion of pre-existing clones that were seen at very low levels of abundance pre-DLI? So for that question, we collected paired TCR and RNA-seq data from two of our responders. And we found that the most dominant clonotypes seen 4 weeks after treatment, for example, in this responder, has a strong overlap with the dominant and expanding clonotypes pre-DLI for the same patient, and none of them were coming from the DLI product. In fact, we only saw overlaps in some of the very low frequency clonotypes with the DLI product, which is the treatment itself. And some of these pre-existing clones, we also see them still at the dominant in 3 years after the treatment as well. Sorry, one is missing here. It's just 144 weeks. So There's also more supporting evidence about the relationship between the terminal exhausted and progenitor exhausted subsets, because when we looked at the clonality of the TCR data, we found a significant overlap between these two states compared to overlaps with any other transcriptional states. Additionally, we see much greater diversity in the progenitor exhausted subsets than the terminal exhausted subsets, showing there could be some selection after differentiation. So to summarize, we found using common factor analysis methods that there are multiple forms of T-cell dysfunction, especially seen in non-responders attributed to resistance. And we found that using the phenotypic volume metric, the pre-treatment T-cell diversity could be important for the response. And the response mechanism involves two major subsets of T-cells, the CD8 positive terminal exhausted subsets that are enriched pre-DLI. And we see that the reversal of T-cell exhaustion is associated with shifts in the cell type composition, which we captured with the Gaussian process regression models. And they also have linked regulatory mechanisms that we identified with Symphony. We also saw that the expansion of the progenitor exhausted subsets is not due to their direct introduction from the treatment itself, and is rather expansion of pre-existing clonotypes that DLI is inducing or recruiting novel clonotypes. Last but not least, I'd like to thank my amazing mentor, Donna Pear, multiple members from the lab, especially Cassandra and Zinning, for their help with the computational modeling, and our collaborators at Dana-Farber, Kathy Wu, Pavan especially, patients, our funding sources, and lastly, we have several open positions starting January at Columbia, and please spread the word. Anyone interested, feel free to contact me, and thank you for your attention.